Hello. In this video we're going to be talking about proteins. So let's not waste any of your time. Let's get right to it. Now when we're talking proteins, we're talking about the workers of a cell. If there is a job that needs to be done, if there's a task that needs to be completed, a protein is getting it done. They're the worker bees, they're the pack mules, any reference point you want to make to show the references the workers, that's the proteins. They handle all the duties, every task. If there's a job to be done, as I mentioned, it's a protein. It's getting it done. Now, proteins are polymers, as all these macromolecules are. And just remember that their monomer is amino acids. We're going to put together a long chain of amino acids to form a protein. And there are 20 major amino acids, 10 of them that are essential, that you just have to have. And you take these 20 amino acids, and through just a multitude of combinations, you can make all of the proteins that are out there. And they can be as small as a couple of amino acids to some that have over 5,000 amino acids in length. But there are hundreds and hundreds of different types of proteins. And if you're thinking, well, how can you make so many different proteins with only a few amino acids? Well, just think about a piano. Okay, just think about a piano. I'm not a piano player. I love music, but not musician, not music oriented, not art oriented. But if you think about a piano, and I think it's got 52 keys. I could be wrong about it. As a matter of fact, I probably am wrong about that. But 52 keys, and in essence, the ones that are very, very high aren't used. The ones that are very, very low aren't used as often. Because people's voice range isn't that wide. So if you just take those middle 3/5 of the piano keys, just think of how many millions of songs have been written with those 35 to 40 keys on the piano. So that's what we're talking about with amino acids. You can take a few and make lots of combinations that are varied. So when you take a strand of amino acids and you put it together, and that process, of course, is dehydration synthesis that you build that with water molecule leaving, all that stuff that we've been talking plenty about. But when you do make those chains, that chain is called a polypeptide. Anytime you see a word reference to peptide, you're talking about proteins. You're talking about amino acid chains. So when you put those together, it's called a polypeptide. Now, the bond between the amino acids, just like with the carbohydrates had a special name for when their monomers were bonded together, a glycidic bond. Bonds between amino acids, or amino acids that are bonded together, that bond is called a peptide bond. And again, it's not a type of bond like covalent or ionic. Those are the actual processes that we're talking about. This is just a name given when a bond occurs between or has occurred between two amino acids. But that's called a peptide bond, and it forms long chains of are called a polypeptide bond. Now when we're talking proteins, I mentioned that they do all the work within a cell, and just a few of their functions, and there are plenty more. You could talk a particular, like in a DNA. When it gets broken apart, that's a particular protein. When it gets red, that's another protein. Another protein comes along and makes sure you got the right nucleotides attached. So they've got all kinds of tasks, but some general words that we can use for their functions. enzyme catalysts. Okay, they can be enzymes or proteins. They can be used in defense, transport, support, motion, regulation, storage. All these things these are some of the things that proteins do. Now, when we're talking structure, okay, structure of a protein, the diagram beside me there, you can see the different sections of a protein, and they're numbered, and we'll talk about each of those numbers. But when we're talking that first group, that's that central carbon atom right there in the middle. The arrow is pointing to it. Blacks are carbons, in this case blues are nitrogen, oxygens are red, and the hydrogens are the white or very, very light gray. To the far left, you've got the amino acid group or the amino group. Okay, that's an amino acid. Number 3, the carboxyl group, that's the far right. You get that CO, and then you've got the OH. If it's just an OH by itself, it's a hydroxyl, but when you throw in a carbon as well, now it's a carboxyl group. And in the middle, on the top side, you've got a single hydrogen, and then of course you've got number 5, which is the one that's most important. It's labeled an R, but it's a variable group. In other words, whatever amino acid is put in that spot determines the function of the protein. And the way I talk about this in the classroom, think about fighting robots. Okay, you want to build a robot to compete in duels. And you build 5 robots, and you build them all the same except for the head. Okay, on one robot, you put a flamethrower for the head. On the second robot, you put something that chomps down or bites. On the third robot, you put a spinning cutting wheel. On the fourth robot, you put a laser that can cut. And on the fifth robot, I'm running out of things to... you put a hammer that smashes. Okay, so each of those robots are built the same except for the head, and because of the different tools you put as the head, it determines how that robot is going to attack its opponent or its purpose. Is it going to burn? Is it going to cut? Is it going to smash? Is it going to chomp? And proteins are the same way. Whatever is put in that R group is going to determine its function. And those R groups can be a variety of different amino acids. Now, R groups have all kinds of options. They can be nonpolar. Okay. They can be charged. They can be polar uncharged, which is a very unique situation. So there's all kinds of things that can go there. And again, that determines the function of the protein. Now, we mentioned this earlier, but a peptide bond, okay, we've got a reference point here, again, to dehydration synthesis, as you can see in the top part of the diagram beside me. The top part is the before, the bottom part is the after, and you can see in red the OH on one of the amino acids, but the other amino acid only has an H there. And so that's a slight variation on our attachment points. So it is going to go through dehydration synthesis. It doesn't meet those two OH ones. The actual OH is on the opposite side, ready to attach with the next one. So the dehydration synthesis has some variations. Now it just says, but it does lose water. and no longer is the bonding element a single element that connects the two. Now you can see that the C of one group and the N of the other group is what is connected together. So I wanted to show you this because it does have some slight variation. It does go through dehydration synthesis. It does lose that water molecule, but the attachment point, the attaching element, and the attachment points are a little different process. So I wanted to show that here. Well, that is our look at proteins, some general information about proteins. When we get together next time, we're going to be talking about the 4 structures of a protein. So we'll see you then.